Today we're going to go over a demo of CelloV2, where there have been some demonstrations of CelloV1, but we're going to focus on this, starting with the About page here, where there is a link to CelloV1, along with the papers that support CelloV2 from a publication in Science. There's another publication in Nature as well, where Cello uses genetic circuit design automation to go about and set up Boolean logic gates that are representative of DNA parts inside of a synthetic biology system. Down below, we wanted to thank some of the folks who have worked on Cello throughout time. And as we continue to go down this About portion, there are different repositories that are the underbelly of what CelloV2 really is. These three applications here are the core of Cello. This is what the web app that you see here is produced by, where this command line interface is really the algorithm behind Cello that produces the resulting files. There is the web app that allows you to interact with Cello here, along with the GUI, which works with the REST API to really present all the UI features that you see in the back, along with this little module that shows us the About page. Last things are you can see your versions at the bottom, the GUI, the API is missing, and the core, where this API here, what we're going to do to fix that is simply log out, log back in, and it should fix that issue where it's just a session caching issue. After so long, the session times out and the API refuses to acknowledge that you've logged in and requires you to log back in. To start this off, we're going to go into running Cello on your own local computer. This is because there is a 30-minute timeout on our website, cellocad.org. For long-running processes, we request that you run this on your local, which I will show you now. You go into this CelloV2 web app. From here, there's instructions on how to install this a variety of ways, but one way I prefer to use is here with Docker. So we'll open up a terminal. Hopefully you have Docker already installed. If not, there is a link below to go about and help you figure out how to install it with the instructions from Docker. There's a few things we need to change here, where we're going to do this in a few places where there's a few local paths, and we'll go into explaining it as I finish up changing this last one of exactly what this command line is going to do. Docker allows you to go about and run applications that you wouldn't normally be able to run. Instead of installing Google Chrome or installing Spotify, you would go about and simply utilize Docker to install Chrome or Spotify and not have to download as big of a package, hopefully, or not have to worry about conflicting requirements on your system. So on this command, you have a Docker run, and this RM means to run in the background on port 8080, so we can access it in our browser. And this here are three different volumes that we're going to be utilizing. The first is for the database. The second is for all the users that you're going to be creating and saving. And the last are the project resources, so any of the resulting files that you create. And then this is the tag that we're using to pull from and run. It's the CyderLab Cello web app. So as I hit enter, this might take a minute or two, because what it's going to do is it's going to start to download it, and then after it downloads everything, it's going to start to run it. It's about 1.5 gigs, and so it might take a minute. I'm going to close out of this, and we're going to go into the actual demo, a little bit more of the scientific portion of this, where Cello goes about and utilizes the algorithm to combine three different tabs and combines them to form some results. The first one is library. You can pick any of these. We have a few strains of E. coli, we have yeast, and we have another stomach bacteria down here. I'm going to just choose this first here, E. coli strain, where you can download the library if you'd like, or you can find out more information by clicking that info button, and that will get you a good chunk of background that you might need if you're trying to run more experiments on these, along with some of the initial authors. You can also upload your own UCF file at the very top right. We'll go into after this demo a little bit more of exactly how you would go about and create one of those UCF files along with the additional inputs and outputs. As we go from the library selection, we'll go into the design, where the first thing you'll see is this Verilog editor. You can write Verilog yourself and just go about and write whatever you'd like to write. But we do include some example files here so that you can go about, set up whatever gates that you need and whatever logic. You can update this as well, where Verilog is a lower level computing language, really going into the circuits of your computer. There's the outputs. You can add in another one. Let's say if I wanted to have a C variable, I could add it there, I could add it there, and when I come over here to select the file, C will appear. But we're not trying to increase any of the complexity of this circuit. We'll go from here, we'll select our input. It should be the same as the library we chose previously. If you'd like, you can upload your own input, and you should be able to download the inputs here. If not, we can always refer to the UCF cello repository. So we'll go here, we'll go with the aura sensor, IPTG sensor, we'll do the output down here, and we'll select a reporter. This is pretty much what I want for my circuit, in which it was all pretty much pre-populated. We'll go once again at the very end to discuss a little bit of how these input files are generated and output files are generated, which is quite similar to the UCF. As we go into our settings, these are the last few things we'll need to change. There's the DNA compiler, which is just the back-end cello that is running to go about and run all this logic. Similarly, Yosis is for our Verilog. As you go, each of these settings is described extensively in the papers, but we won't go into each and every one of them, just some of the more important ones, where I would say some of the more important ones are the open source repositories we're using and utilizing in the synthetic biology community, such as Eugene, which is an open source compiler that is based off open source language of Pigeon that helps you generate S-bol diagrams and other circuitry. Similarly, going a few layers down, there's this export of S-bol, which is the synthetic biology open language. It's the standard that really helps us all get on the same page of how we're going to communicate across the world how genetic circuits and how genetic systems can work. This export of S-bol is compatible with another open source registry known as SynBioHub. SynBioHub here is very useful, as it can go about and you can pretty much share any of these cello designs that you're creating with coworkers, along with go about and share it with the world. And then lastly, they have a collection URI, but as you get into actual generating these results, it will give you the option of logging into SynBioHub or whatever SynBioHub you want, and you can go from there to go about and set up anything you need. You can also change that repository URL because there are multiple SynBioHubs out there. As we're here, we're about to hit synthesize. It's going to ask us to generate a project name and description, but I'm opening up this developer console in my browser just to try to keep track of some of the time. It's a little bit more advanced. You don't have to do this part. But from here, we're clicking the start button. We're going to call our project name the Demo Project V1 for CelloV2. And what we did is, I think we did an IPTG sensors plus a YFP reporter. I'm going to hit submit. This will help us track some of the time it takes for all this to go through. It can take, as I said before, we allow up to 30 minutes for your job. If it's going to take longer than that, I ask that you run this on your local, which we'll get back to right now. It's finished downloading, and so I can actually go up now and it's running too, as you can see by that big old cello right here in the center. It's starting the application, so give it a second to load up. And while that's happening, we didn't miss anything on this side. And there we go though. I'm already logged in here, but it's gonna be working just like cello is up on cellocat.org. But it looks like I have another cached issue, so I have to log out and log back in. Test. Okay, close. Never. We don't want to save the passwords right now. We'll go down and we'll see that that should be okay. If I hit the plus, you see all these libraries have produced now. Where previously we went here and it said no data to display. If I go into the about, it says the API is up, but that log out and log back in seems to have fixed it. As we don't need to rerun this again, you see that project demo v1 was actually produced successfully. We're going to now go into some of the results where it will show you the same circuit in different ways. So all these are synonymous with each other. They could all be implemented and they could produce different results. This is not a real rocket science. This is a little bit more biological. So not everything's down and exact. I believe in the Nature paper, when they went and tested all the different variations of a circuit, 45 out of 60 produced what would eventually become real logic or real systems functioning as expected, where A and B produced a primary output, for example. And as we get deeper, this is your logic sort of diagram, and as you get deeper into it, you'll see some of your transcriptional activity where the output here is a combination of A and B, in which I would expect A and B to not have 100% output, and there should be a little bit of loss. Similarly, these are your promoters here, your promoter circuits, and what they should be representing. And as we go down, there's a truth table, so you should know exactly what should be producing your YFP promoter, in which A and B will have to be together and both present. If you don't know too much about truth tables, it may be something to find a supplementary or additional video, similarly with Verilog, or even more on the synthetic biology side if you're wondering more about how these different promoters work, then it may be time for a little bit deeper of a science lesson. As we go down into this last portion, what this table is representing is a bit of the response plots, where the filled in circle means that the promoter is present, and the empty circle means the promoter is not present. Just so you're able to read these diagrams, and we're going to go deeper as we go into placement. Like I told you before, these placements are all synonymous from the very top. They can all be utilized, and some might be more effective than others. Lastly, the export. If you have a SynBioHub account, you can log in. I didn't really talk too much about SynBioHub, but it is a registry, and it was started up, I believe, by Chris Meyer's lab. He's out from the University of Colorado Boulder, where he works in synthetic biology and doing some of these circuit design automation along with other applications, especially in SBOR, where SynBioHub will actually allow you to export at the end of the day to other standards like GenBank and FASTA. As you can see inside of Cello, we have multiple SynBioHubs, since this is an open source application as well, similar to Cello. It means anyone can edit it, anyone can set it up. There's one for Utah, there's one for this programming biology, and there's synbiohub.org, of course, and there's one for the Cider Lab. all of these might have different parts and different portions of the registry you can utilize. If you don't find what you're looking for in one registry, you might be able to find it in another. So I do implore you to look around a little bit to find the exact registry you want. In minutes or so, I'm going to go over the generation of UCF input and output files for CelloV2, where you can go into genetic circuit design automation with Cello 2.0, and Nature has that paper, which has supplementary information by Nielsen et al., Alec Nielsen, where the main paper for Nature was written by Timothy Jones et al., and it really goes into things of how these files work. There is a little bit of the original version of Cello, which provides this for free if you don't have access to Nature. Under the About page, you go to Supplementary Info, and it will give you the document that you would need to go into every single part of any of these files to really generate them. It will go a little bit into Cello, go into Verilog, but under section 7, you can see format of the user constraint file on page 58, along with some of the materials, methods, and what these different sections really mean. So you always have that, but I'll go into it for now, where all these are pretty much required fields except for the date and the author. Those are optional. You're going to need a version, where the version can be a variety of things, where right now we have this sort of organism name, but it could be, instead of just having this, you could definitely use an organism ID. You can utilize a few other types of fields as well if you want to use a number or some strain identifier, experimental condition identifier, maybe genetic gates, or even technology mapping or motif identifiers. You can utilize those. Just know that the date and author aren't required. The organism, genome, media, temperature, and growth will need to be here in one form or another. Where I think they're a little bit self-explanatory if you have some sort of science background. And I'll go into some of those fields that aren't as self-explanatory. From here, we're going to the measurement standard, measurement underscore STD, which all three fields here are needed for the signal carrier units, the normalization instructions, plasmid description. And in my, this article here from Nature, it says that we need a plasmid sequence as well, but it's been omitted from here, so I'm going to guess not necessarily needed. From here, you go into some of the genetic locations. This is where these fields will actually be, or these genes or promoters should actually be located on your plasmid or genome, where this is your symbol, L1, you have your description, there's the locus and the sequence, where you can get the sequence from a file, or you can use an NCBI identifier, where this only has these NCBI identifiers, and it's probably the better option to use. As you go into the next section, those logic constraints are a way that you can go about and really sort of say what comes before what for your sensors, your promoters, and what's going to be expressed. So we have these two for NOR and the output OR, where it tells you how many instances it can have at max, and you'll need both of these fields to go about and generate this. The outputs in this example only has one, but it has up to 3 inputs, and it lets you know exactly how you're going to wire these up with some Verilog below, where a wire is supposed to be sort of a connection between two different gates. As we start to get into these motif libraries, these have outputs, inputs, and net lists. And what you'll do with those will be pretty much hooking up everything like I previously discussed for the logic constraints. There are some more constraints that will come at the end, but for now we're going to skip a lot of these motif libraries because a good chunk of this file is just that. Motif libraries, we're going to go maybe another couple thousand lines in, And you have plenty of examples of motif libraries. This is why it takes so long to really get through this and have somebody generate a new organism for going through cello. It is an adventure to say the least, and I admire anyone who can go through this from both the technical perspective and the scientific perspective. But we went a little bit too low, it seems, as we passed up these motif libraries and we started to hit structures. So I can just discuss these structures as we've hit them. I wanted to make sure we didn't miss any sections before the structures started to appear, where we were scrolling a little fast. But we did, you see, we missed the gate section right after motif library. So these gates are going to be a way of going about and just essentially establishing what you're going to, I guess, at the end of the day, these gates are going to be the promoters. These are going to be the actual organisms or the DNA sections that you express, whether it's more likely a sensor than anything. But you'll need to start off with the name of the gate, and then the system is really the biochemistry behind it, where TEDR is a very common promoter that you can use in E. coli. From there, you get the group, which is like a set of gate variants that you could potentially be using. See, this is the name's P1 for CelloV, but the group is just the CelloV. So there could be a P2, there could be a P3 out there, but we're not going to really look into it. And then we go into what kind of gate type this is, where a NOR gate can be implemented, and you can use that NOR gate to implement a NOT gate. The only allowed values here are certain Verilog values, so NOR and OR, output OR, where it's going to be sort of like an output underscore OR until output OR, just like that. And that could be a gate type. But it's standard Verilog, I would say. You could also have NAND, XOR, and XNOR gates. But we won't go into each of these gates here. Although they're all the different A, B, C that we had previously mentioned on these motifs above for inputs, these are most likely the gates. Where TEDR has a few different names, even though it's all the same system. Now we go into structures, where structures have inputs, outputs, and devices. Where in your inputs, you will be required to have a name and a part type. Most of these are going to be promoters at the end of the day. Your outputs are going to be your outputs, your actual resulting, where these are just the names are N1 and N2 and what you want for your actual representation at the end of the day. Lastly, these devices are actually parts, where the components are compiled of a multitude of parts, and you'll get a variety of different parts from it. But let's say this one feeds into this one. You can look at this one feeds into up here for CelloFA, and if I look up for CelloFA, we can find it inside of our gates. So it all does kind of hook up together. Make sure you don't drop anything necessarily in these relationships. But we'll go back down to our structures right below the gates, and we'll let some of these, we'll go through, pass up these gates, because as you start to read through these, you'll see them as pretty standard. They'll all need components, they'll all need a name. And then I think we're going into some of our models next. These models are going to be how you do some of the calculations, along with getting into eventually the specificity and toxicity, where we'll just grab this model here for P1 CelloV model. It has a response function, hill response, which we'll look at later, but it does feed in some of the input composition as well, which we could look into in a second. along with toxicity and cytometry, which you'll need to act if you are designing this new library. You'll need to go and run those experiments to really get the numbers behind it. What's the Y max here? The maximal transcription, the minimal transcription, and these other variables as well, which will feed into this Hill response function. The only thing that's not required out of all these things are descriptions for your parameters. Everything else you'll need to keep, it's required. And we'll go down now into some of the functions I want to say. All of these are going to use Hill response in the linear input composition. And as we get to it, I think that's the next section we're about to hit. Yep, here's the Hill response into functions. See the equation with all the variables from the above. And then we can go along with the parameters, where you're going to need the name and the map, and the names and the maps on all the parameters along with the variables. It just helps you describe exactly what you're going to be grabbing. As we go, this is also a linear input composition, which we saw above. It's simply doing an equation of x1 plus x2. And lastly, what we're getting into are our circuit rules and device rules. This is similar to some of the logic we had to implement before above, where it would say like an AND gate. And what does that AND gate to? As you go from L1, it has to be next to L2, and L2 has to be before L3, where eventually you'll have some reporter that comes after it all. Now, this is just a little bit of the logic along with how all the different parts are going to be interacting. If they're going to be facing forward, if they're going to be facing backwards, but you might not need to utilize the backwards facing arrow. So, most likely you'll see all forward. This is when we get into the parts really. Parts have three things that are required. It is the type, the name, and the DNA sequence. You have all three of those for every one of your parts. You'll be all set. I do believe a good chunk of the rest of this document is parts until you get to some more functions and how these functions are being calculating for mainly toxicity and cytometry. This is where, like I previously said, you're going to have to run these experiments yourself and get the results for this table and what the output would be. All of this for the rest of this document is toxicity and cytometry. It's about 30,000 lines of it, where we were just up at 10,000. As I go to the bottom, we hit about 45,000, almost 50,000. I'd like to thank you all for viewing this. Please leave any comments or questions below. If you'd like to support Nona Research Foundation, in which we host this, along with a variety of other open source software tools, please feel free to donate, and we'd love to have some help in hosting these servers. Thanks.